Welcome to the Neural Implant Podcast, where we talk with the people behind the current events and breakthroughs in brain implants in an understandable way, helping bring together various fields involved in neuroprosthetics. Here is your host, Ladan.
Hello, everyone, and welcome to the Neural Implant Podcast. Today's guest is Mauro Gandolfo, and the reason I'm saying it like this and having so much fun with it is because he is Italian, and he works for 3Brain, which is a large-scale sensing microelectrode array platform for label-free in vitro electrophysiology. And they are based in Zurich, Switzerland. Basically, these are ways to get organoids and brain slices into a device and be able to image them with very, very high resolution, both spatially and temporally, time-wise, and they're able to have 4,096 channels, CMOS-based technology, and 18 kilohertz, which is, oversampling, but, compared to the 1 kilohertz that we like to hear about in neurons. I thought this was really cool. I'm all about devices. I'm all about oversampling. I think better always to oversample than to undersample. I really enjoyed this. Yeah, I think this will be a friend of the show. And again, this is a little bit different than what we usually talk about, implantable devices. As I said in the interview, usually we put the device into the brain, but in this case, you're putting the brain into the device. Ooh, think about that. Okay, hopefully you enjoyed the show. Mauro Gandolfo, pleasure to have you on the show. What's your role? You're at 3Brain. Oh, shoot. What is it? 3Brain. I saw something before. 3Brain Neuro? Or no, wait.
3Brain, yeah. Just 3Brain.
AG.
Yeah.
That's
correct.
Aktiengesellschaft, right? That's like
correct.
A corporation.
Here in Switzerland and in German-speaking countries. Yeah.
Cool. Well, anyways, pleasure to have you on the show. Yeah, you guys have a really cool technology. And as we were talking before the recording, I saw you guys at SFN, and it's essentially, I'm going to say, oversampled microelectrode array that's used for in vitro samples. And you guys have 4,096 CMOS sensors that sampled 18 kilohertz. And basically, you're able to see what's going on inside of organoids or slices or something like this. Do you want to introduce yourself and talk a little bit about this technology?
Yes, sure. I'm CEO of 3Brain, and I'm a co-founder together with other two colleagues. We have been working to this technology more than 15 years now. We were at the top leading European institutes such as EPFL and the Swiss Center for Electronics and Microtechnology here in Switzerland, and the Università of Genova and the Institute. We met each other more than 15 years ago. We started with a European project, and at the time there were already microelectrode arrays, and they were made by passive wires integrated between 8 and 60 electrodes that you were able to use to record signals from neuronal cells. And we decided that we should have improved this, and we started to work on this CMOS technology, which allows us to integrate, as you said, up to 4,096 electrodes for the moment in the same device. And it's made by tiny microelectrodes. And we have, of course, a lot of circuitry underneath and many transistors. And we integrated this microchip at the bottom of a well. The well is like a sort of petri dish, the one that is used for culturing cells in vitro. And you can place cells in this dish, and you can add nutrients and media, and you see the cells growing, and the neurons starts to connect each other to form a network, expressing electrical activity, very similar to the activity that we have in our brain. And the cells are in contact with the microchip, very close to the cells. And in this way, we are able to record spikes from the cells, which are basically action potential, and we basically listen to what the cells are telling each other, and we can also write information to the cells by stimulating electrically the cells and see the response to stimuli. It can be used with different kinds of samples. I was mentioning before a cell culture, but as you said, also brain slices taken from mice, or more recently we are working with brain organoids, which is a very cool technology. It holds a lot of potential for medicine and for finding new treatments for brain diseases.
Yeah, I think so. I think we'll get to that later. So you guys use CMOS technology, and what they use usually in other sensors are kind of more, as you're saying, glass or metal coatings and stuff like this. First of all, what is CMOS? Kind of a refresher for some people who may not remember. And then what's the advantage of that?
I mean, CMOS is a silicon-based technology which is used inside the CPU, and it allows to integrate circuitries in a very small piece of hardware. And we use in particular in our CMOS technology an approach which has been used for years in camera, no? Like the one that you have in your photo camera or the one that you have in a mobile phone, where each pixel comprises circuitry underneath the pixel in order to precondition and process the data. The difference here is that instead of processing photons, we have modified the structure of the pixel in order to record and to sense voltage changes, which happen at the level of the membrane of neurons or of any other kind of electrogenic cells. Yeah, so it's a technology that nowadays it's really used in a lot of the consumer products, and you find in every electronic devices. And we have been one of the first using it for biological applications, meaning especially for brain, we've been one of the first using for this kind of application.
Yeah, definitely. And so what's the advantage of that over some other technology?
The main advantage is that we, with respect to other microelectrode arrays, which are lower resolutions, that here we can integrate a lot of electrodes, a lot of sensors in a tiny area. And as you know, the brain is very complex, and also even a culture in vitro is made by thousands or even tens of thousands of neurons. And if you try to sense these neurons with only a few electrodes, you have a strong undersampling, and you cannot capture what is going on really in your cell culture. Our approach instead, you have thousands of electrodes, and you can sense therefore thousands of neurons, and you have a better idea of what is going on. You are much more sensible. For instance, you can add compounds and drugs that you want to test, for instance, for toxicity studies, and you can better track what's going on, and these compounds and these drugs affect the cell culture. The fact that it's a label-free technology, so differently from other technologies that are used in vitro, we don't use any dyes to record the activity. Just to make a comparison with other technologies like microscopy, in that case you have the cells in your well, and then you observe the cells from the top with an optical system with lenses and a camera connected to lenses. And in order to capture what is going on, you need to colorize basically the cells with dyes, by making use of fluorescence dyes, which is called also labeling the cells. While with our approach, by having the CMOS contact with the cells, we don't need to use any kind of dye, and we still obtain a sort of imaging of your cells, but we call it functional imaging because we see how the cells behave and how they interact between each other by communicating these electrical pulses, these action potentials. So we have a functional information of what is going on your cell cultures. There are also many other advantages. One that is also the fact that we can observe over time what is going on your cells, no? And this is particularly interesting for neurodegenerative diseases or for developmental diseases where you need to observe the onset of a certain disease. Since we are label-free and somehow we are non-invasive, no, for the cells, we don't disturb them, we don't kill the cells over time. We can observe how they behave over a long period, days, weeks, or even months.
Yeah, that's really interesting. And you were saying that the density of the sensors, the CMOS, is much tighter than you can get with using other methods. What is your pitch length? Like, how far apart are those electrical sensors?
The lowest is 40 microns, and we go up to 80 microns. The number of electrodes is still 4096, so we change the density to, in one case, when we go to 40 microns, we observe an area of 2 1.6 by 2.6 millimeters. When we go to 80 microns pitch, the resolution decreases, but we observe a larger area, up to 5 by 5 millimeters, which can be interesting for a certain kind of biological samples where you want to observe both the hippocampal structures and maybe also the cortex at the same time, and you want to measure them at the same time. So it might be interesting to observe a larger portion of your samples.
Yeah, that's really interesting. I remember hearing that basically electrodes can sense neurons from up to, I think it was 50 microns away. So basically that's, 100 micron diameter. You guys have basically 2.5 sensors in that diameter length. So you're essentially oversampling. Is that something that's bad? I've heard arguments that, it doesn't make sense to oversample. You're just kind of wasting data. What is your guys' opinion on that?
Our opinion is that we can increase the information that you can get on your cell cultures. We have not so high oversampling because it depends also how the electrodes are shaped, and ours are squared and they are slightly sunk, so recessed below the planar level of the chip. And typically from one electrode you can sense either one cell, maybe 2, maximum 3 cells. So there is still some oversampling, but that is useful because, for instance, you can use triangulation to understand where is the source of the information, where is the placed the neuron. If you can sense the same neuron with 2 or maybe 3 different electrodes, and in these electrodes you have different amplitude of the signal coming from that neuron, then you can localize where the neuron is in place, and this helps to understand the topology, for instance, of your network. Additionally, having more electrodes, you can also observe how the signal is transmitted across, for instance, the axon of the neuron. And so you can measure the velocity, the speed of the signal going through an axon. So overall, I would say that you get more information. And I think that our is not an extreme oversampling, and it's a good compromise between the resolution. So we still keep an eye on the spatial resolution, but on the other side, we also try to go over measuring a large scale of neurons, no? Because as I was mentioning before, we go up to 5 by 5 millimeters. And the properties of a neuronal network in our brain, but also in vitro, they arise at the level of the network, no? So, and it's important to capture a large network in order to understand what's going on in your preparation.
Yeah, definitely. I mean, when I saw you guys' booth demonstration at SFN, Society for Neuroscience, I was impressed. I mean, it is slightly oversampled, but because you have these kind of pixels almost, you can see as the action potential is going across, the screen and how it's actually going in this two-dimensional thing. And it's just amazing. I mean, even if it does take more data, better to oversample. And then later on down the line, you can, delete half of that if you want, or if you're really, convinced that you don't need all that data, just turn off half the channels, or 75% of the channels if you're really convinced that you don't need that, and just don't use it.
Exactly. Indeed. Our users are quite free on that. They have compression tools, and they can decide the level of information that they want to capture. And depends also on the questions that you have, what you are searching for, which are your hypotheses, and what you want to test.
Yeah, definitely. So I want to transfer now. We haven't really talked about on this show, Neural Implant Podcast, we talk more about in vivo, implants, putting something into that. So putting some kind of device into the tissue. But in this case, you're putting the tissue into the device. So do you want to talk a little bit about in vitro tests and organoids? Actually, I don't know if we've even talked about organoids on this podcast. Do you want to explain all this?
Yeah, then it's a good opportunity to introduce to your audience. So our purpose is to make what we call a brain-on-chip device that can be useful for preclinical studies and drug discovery. So as you know, when you want to study a new compound, a new drug for a certain disease, you have first to perform studies in vitro before you can go to clinical trials and test on humans. And the brain has been always quite a difficult organ to study for several reasons, but mainly because the researchers have been left with little material to study. You cannot simply drill an hole in the head of someone to take a sample. And also, if I would ask someone to donate a sample, I don't think we would find much participant on this.
Most people are attached to their brains, huh?
Exactly. And so because of this, the research for the brain has been always lagging. Maybe an interesting thing is that, Alzheimer is a very painful disease that for which we don't have any cure. But maybe not many people know that Alzheimer has been cured hundreds of times, but only in mice. Okay, mice is just a model of the Alzheimer, so it's not exactly the same that we have on humans. This still gives an indication of the fact that if you have the material to study, you can achieve your goals. But since the brain and the human brain has been so secret somehow for researchers, it has been always a problem. More recently, there is a revolutionary, I would say, breakthrough technology that is going to reshape medicine in the 21st century. And that is the stem cell technology and the cell reprogramming technology. Thanks to that, nowadays it's possible to take a sample, like a biopsy, from the skin of a patient and to obtain stem cells, which are induced stem cells from that sample. And then with the right ingredients, which are the right factors, growing factors and compounds, you can reprogram the stem cells and you can make neuronal cells out of those stem cells. And the advantage is that you have the genetic profile of the patient. And therefore, you can also capturing the disease profile of the patient in a model, let's say, of the brain. These cells forming, they can aggregate themselves with the proper protocol, and they can recreate a tiny brain, which is called the cerebral organoid, which is at the moment very small. It's just what researchers can do is just a very simple, a very simplified version of our brain. And it's not, of course, containing all the areas that we have in our brain, but still is a three-dimensional structure, smaller than a cherry seed. And the researchers with this, they have finally human cells, and they can see the development of the brain organoid over time. So they can study it, and this gives all the tremendous potential for treating in the future brain diseases like Alzheimer, Parkinson, autism, epilepsy, and many others. So what we do is we provide these researchers with a technology that can help in studying and understanding these brain organoids. By placing these brain samples on top of our chip, they can observe what's going on in these brain organoids, and they can maybe test compounds that could help to treat a certain disease, and they can see whether the compound has a positive effect and is able to restore the normal condition of a brain sample. And the opposite, as you were saying, of what is done in vivo. Here we are taking the brain and we are placing on top of our chip.
Yeah, definitely. That's pretty interesting stuff. I'm also curious, how accurate is this? I mean, how much of a proxy or a replacement is this for in vivo stuff? Does it translate well, or are the in vitro things completely different? Because, I mean, you can imagine a lot of the biological processes aren't there in slices and organoids, blood flowing through, and I don't know what else, but how accurate is it?
Yeah, I mean, brain organoids is a technology that is under development right now, and researchers, they are keeping on improving it, and we will still need years to have a better model. But we can say that it's already much better than the 2D, two-dimensional models that were used in the past, because 2D culture doesn't recapitulate what is in our brain. It lacks the mature complexity of human organ tissue. For instance, a cell in vivo is three-dimensional, while if you culture a 2D cell, 2D culture, you have the cells tend to be flattened, or even the interconnection between cells in 2D are not the same that we have in 3D. And also the diffusion of a compound is not the same. Three-dimensional organoids are already much better than what we had in the past. Then, of course, researchers are working on improving them even more, on adding vascularization, on integrating more kind of cells inside these cerebral organoid, because you have to, for instance, trying to recapitulate cortex and hippocampus and different kind of structures as we have in our brain. And this is a work under development.
Okay. Cool. And then you guys have 4,000 channels. What's stopping you from doing more?
We could do indeed more, but where we are focusing now is on other two kind of developments. One is to provide researchers with higher throughput devices where you copy and paste the well I was mentioning before with our chip in multiple replicas. So instead of having a single well, you have maybe 6, 24, or even 96 wells. And these can test more compounds at the same time and at different concentrations. So we are not integrating more electrodes in the same chip, but we are going to integrating in the same plate, in the same device, more chips, which at the end, if you imagine, if you 1,000 electrodes per well and you multiply by 96 wells, which is a standard multi-well plate, you obtain almost 100,000 electrodes, which is still something really remarkable in terms of data management. So this is one of our developments that we are working on, and the other one is to go three-dimensional, because as we have now brain organoids which are three-dimensional, we really believe that we also need a technology that is three-dimensional and that can help studying the brain organoids from the inside, so penetrating inside them and recording activity from inside the brain organoids. These are our main two development focus at the moment, and we are running them in parallel in order to provide to researchers a more powerful technology. We think this is the direction to go.
Yeah, that's a good point. I mean, probably they don't need bigger organoids or bigger sample or more dense. Running the experiments in parallel is probably the much more beneficial thing for them. That is pretty interesting. And then another thing, you guys run the samples at 18 kilohertz, but I thought that neurons max out at 1,000. So why again the oversampling?
You need a higher sampling frequency in order to perform, if you want to do spike sorting, which is a technique that allows you to distinguish the different neuronal sources that you can pick up with the same electrode. I mean, typically you need around 12,000 sampling frequency at least in order to being able to sort the spikes. So to identify from the same electrode, which is picking up signals maybe from 2 or 3 cells. If you want to identify which is the signal coming from one cell and which is the signal coming from the other cell, you need to have at least around 12,000 samples per second. And we can go up to 18,000, but indeed only for some specific applications. And we have, you can also, our users can adapt that to their needs, and they can go down to 15 and 12 or even 10,000, depending on the kind of applications and the kind of test they are working on.
Wow. That's fascinating stuff. I think this is really cool. What kind of researchers are you targeting? And then what kind of researchers do you hope will start using this technology more?
We are working with a series of researchers that are focusing on different kind of diseases and different kind of brain models, going from neuronal networks, from cell cultures to brain slices to retina. We have also users and researchers working with retina. And what we target more now is brain organoids because we think that this is really interesting and it can really help the people to find new treatments. And we would like to have more users in that field in the future. And we are convinced that we will be able to do that once we will have this new 3D technology that we are working on, which we believe the perfect match for this kind of samples and users.
Yeah, but with that 3D technology, I mean, you're limited basically with semiconductor processes, and those usually can't get too high. What kind of height difference are you guys expecting or hoping to get?
Yeah, indeed, is post-processing. And at the moment, we can achieve an height of our silicon chip where we deposit on top structures, which are gold structures, 100 microns from the surface of the chip. And maybe in the future we could go even further. But 100 microns is already a good size, because when you have a brain slice or a brain organoids, which is maybe 400 microns thick or half a millimeter thick, when you have 100 microns, you can already get inside and try to understand what's going on. So we are working now to improving even more, and we hope maybe to, if it will be needed also by researchers, we will always carefully listen to their needs, but we try to go even further. But at the moment, our goal is around 100 microns. Yeah.
Wow. Very cool. Well, Mauro, this has been very interesting. I've really enjoyed this. I mean, again, this is a little bit more up my alley. I like the hardware side of things, and I love oversampling. I love this kind of stuff. Can we expect to see this one day in vivo implantable stuff?
Well, it's not in our plans to go in vivo, not for the moment at least. But I know of other companies that are working in that direction with still silicon-based chips or with different kind of approaches, and I'm sure you will see more and more also in vivo for implants for similar technologies to the one that we have.
Yeah, excellent. Is there anything that we didn't cover that you wanted to mention?
No, I mean, it's all good. You allowed me to give a good overview of the technology and of what we do and our plans, and yeah, I mean, maybe just as an ending note, I mean, I would like to mention that we are driven by one mission, which is providing technology in order to try to help researchers to find new cures for brain diseases. And brain disorders are very broad, and there are more than 600 conditions touching 2 billion people, because they count in addition to Alzheimer, Parkinson, but also sleep disorders, addiction, depression, many other conditions. And we work for that, for millions of people with brain impairments, and also for their loved ones, as there are several diseases that are very awful also for relatives. So that is our mission, and we hope to give at least a little contribution in that direction.
Yeah, for sure. And then we're recording this in the middle of the coronavirus epidemic. Any plans to help with that?
We are still keeping on our focus on brain. There will be the need in the future also for brain disease. The prevalence is increasing because we are also living longer. And for the moment we focus on that. I think it's quite already challenging that, and we stay on that.
Yeah. Yeah, well, maybe one day. Anyways, Mauro, this has been excellent. Thank you so much.
Thank you so much, Ladan. It was a pleasure to talk to you.
Hey guys, what did you think about that? I really enjoyed it. As I said in the intro, I really loved devices and oversampling, having way more data than you need that you can eventually throw out if you want. That's better than the alternative, I think. Anyways, hopefully you guys enjoyed it as well, and, are not too mad that it's in vivo style stuff. And, I think we can do in vitro style stuff, even if we have in vivo experiments. So, yeah, I really enjoyed this. I think this is excellent stuff to look at. He did offer to give more names about people who do in vitro stuff, and I will definitely reach out to them, because I think this is interesting stuff, especially organoids. It's cool. It really is. So hopefully I'll be able to prove that to you in the future. Anyways, let me know what you think. Write to me. Let me know what you think in a iTunes review. Hopefully it'll be accompanied with 5 stars, but if it's not, that's okay. Okay, see you guys next time.
Hope you enjoyed the show and were able to learn something new, bringing together different fields in novel ways. Until next time on the Neural Implant Podcast.